Hi, welcome to the Brain Fix podcast, discussing all things brain. I'm Professor David Brough, Professor of Neuroinflammation at the University of Manchester, and the first guest on the podcast is Professor of Neuroscience Richard Baines, and today we'll be discussing the use of fruit flies in neuroscience research. So Richard, welcome to the podcast. I wonder if we could start with a little bit of background into your research career and what's brought you to your current research interests.
Well, thank you, David, for inviting me. My background has always been a neuroscientist, but one that's always worked with insects. And insects have a very special property in that because their nervous systems are quite small, they have what we call identified neurons. That means we can actually give names to specific nerve cells and go back to the same cell time after time in different insects. You can't do that in higher animals like mice or rats and so on. So it gives us a level of resolution that's better than competing models. Now, I started out with what we call big insects. So I finished my PhD back in 1988, which is rather too long ago for my liking, actually. But I worked with locusts and I worked with cockroaches. They had big nerve cells, but they had no genetics. So in about the 90s, when the molecular biology genetics revolution really got going, I made the switch to fruit flies, Drosophila. Much smaller, but still have identified neurons, but now they had the capability to mix genetics and molecular biology. And together it's now a very powerful laboratory model system.
Yeah, so I can see that, mixing the genetics makes a massive advance, but I guess how similar is a fly brain to a human brain, and what can you actually model with it?
Well, I get that question a lot, and my first response is always to say there's only one nervous system on the planet. We all share a variation of it. So if you think about a fly, it's a lot smaller, and yes, its nervous system is a lot smaller. But flies still need to do everything that we do. So they need to be able to see, they need to be able to hear, they need to be able to walk, and in the case of a fly, they fly, something we don't. They find food, they need to find a mate. So actually, when you boil it down, the fly is not so different to the human. Now, if you take a fly and look inside and find the nervous system, what you will find inside that nervous system is very recognizable from a human perspective. The nerve cells use the same sorts of neurotransmitters, the chemical signals. They have the same sort of basic structure. The connections between them, which we call synapses, are the same. And so in many ways, a good analogy is perhaps thinking about a laptop computer, being the insect, and a supercomputer that might take up an Amazon-sized warehouse as a human brain. It's a matter of scale that's different, but not the actual functionality of it. And the advantage for that, for us really then, is that we have a nervous system which is composed of much fewer nerve cells, maybe 1 million nerve cells in the fly brain, whereas a human has 80 billion. We have nerve cells which we can identify and go back to, and we have nerve cells that we can manipulate the genetics of.
So in terms of your current field, your current research interests, I know you've done a lot of research on epilepsy and trying to find new ways of treating it. How useful has the fly been in this area?
So in many ways, the issue with epilepsy is it's a very common neurological disease. So 1 to 2% of the world's population suffer from epilepsy. So in the UK, that's 600,000 people. Now, we have a lot of drugs in the clinic. There's about 25 or more drugs been developed over about the last 70 or 80 years. But there are two key problems. The first is about 30% of people with epilepsy don't respond to the drugs we have. And even though we keep designing new drugs, that number doesn't change. The second is those drugs are not a cure. They will prevent or reduce seizure severity, seizure frequency, but the individual has to take those drugs for life. And as we know, drugs come with side effects. And with epilepsy, those side effects can be very significant, particularly for women of childbearing age. They have very bad effects on the developing fetus, to the point where women often have to make a very difficult decision to come off medication whilst pregnant, but then they lose management of their seizures. So we need new approaches, new ideas, and that's where things like the fruit fly come in. It's a laboratory test tube to allow us to experiment. So a couple of areas that we've really explored in my laboratory are one, how we could possibly design treatments for new targets. And the second, which I'll explain in a moment, is we've actually shown it is possible to cure epilepsy. So the first one, if you imagine the human brain, as I mentioned before, 80 billion cells, they all have about 1,000 connections each. It's something like 1 trillion connections in the brain. And they're changing all of the time. So actually the brain is a real chaotic place to be. It's not static by any means. And the brain has developed these sort of mechanisms called neuronal homeostasis to maintain activity within acceptable limits. And that's really been an area I've studied for 20 years now. How do these mechanisms work? And we realized some time ago that these mechanisms that the brain already uses to make sure most of us don't have seizures, could we boost them in people who did have seizures and epilepsy for a cure, for a treatment? And so, long story short, we've hijacked one of these systems. We've developed a drug that promotes the brain's own mechanism. And when we've tested this compound in both fly seizure models and mouse seizure models, it's really very effective. And so we've got a new drug target to develop, and it's early days, but it's quite promising. So coming back to that second area we've developed, the sort of holy grail, if you like, and that's curing epilepsy. Could we ever do that one day? So flies have the same genes as humans, and we can mutate genes in the fly, which we find are mutated in people with epilepsy. So when we do that, we end up with flies, we can induce seizures in them. But we found actually that even if those flies have those mutations, if we manipulate the nervous system early in development, when the nervous system is forming, we can actually sort of overcome the effect of the mutation. So the fly develops with the mutation, but never shows any seizures, because we've allowed the nervous system to compensate. So that's quite an ambition. It has recently been reproduced in a mouse model, which is always the next step up the translational pipeline. But at least in the laboratory, it is possible to prevent the epilepsy process. And that really is, where we would want to work towards, because then you take the reliance away from drugs. So the fly is never going to be a human. It's too far apart from it, evolutionarily speaking. But it's a real great test bed where we can generate new ideas and tinker around. How do things work? We know that, and maybe we can intervene.
Yes, and those two examples are incredibly exciting. So fingers crossed that the future's really bright there. More generally, aside from epilepsy, are there targets or treatments for medical conditions that have been first discovered in the fly?
Yeah, I think that's a harder thing to pin down. And in many ways, I think I could say yes, but I can't put my finger on the specifics. And let me explain why I think that is. So almost all that we know about human biology comes from studies in simpler animals like fish, frogs, flies, locusts, and so on. But before we can get them to the clinic in terms of new drugs, they have to go through preclinical testing and preclinical experimentation. And the gap between the sort of early stage and the late stages can be 10, 15, 20 years. And so in many ways in science, the sort of origins of stories get lost. So we all stand on other people's shoulders, but I think we sort of forget who we're standing on at times, and so it's very difficult to pin it down. But I think, let me perhaps illustrate this with one excellent example, a very pertinent recent example, and that is the biological clock. So we all have a clock that ticks away inside us. It anticipates the alarm clock. It tells our body when lunchtime is and when evening is, and it gets us ready for the different stages of the day. The basic mechanism of the biological clock was found from work in the fly. And people in the clinic now, clinicians, are very interested in using the knowledge of the clock to decide when to give people drugs, because you will respond to the same drug differently at different times of the day. There are also other people looking to see how we could change people's biological clocks for all sorts of reasons. There's a good link to cancer, for example. But, simply jet lag. Imagine if we could have a pill which you could pop to stop jet lag. I'd be very happy about that. And there are people working on it. So basic science feeds the clinical science. And so difficult to say a specific example, but I think it's implicit in all of it.
Okay. So I guess the future for flies and research is quite bright then. What do you think the promise for the future in neuroscience is using fruit flies?
I think flies hold enormous promise. So the fly was the first complex animal to have its genome fully sequenced. The worm, C. elegans, was the first, but it's a very smaller, simpler animal. The Drosophila was in many ways the test for doing the human genome. We also now have a complete wiring diagram of the fly brain. So we know which cell is connected to which cell, and that's a huge effort over the last 20 years. So I think it's fair to say the fly is probably the most well understood animal on the planet. And one of the beauties of having that sort of well characterized system is not only exploring biology, but we can use it as a test bed for developing new methods, new ways to look at cells, new ways to manipulate cells, new ways to move genes around. And it's methods that drive science. So with the advent of the Human Genome Project, we developed lots of different ways to make genetically modified animals. And that allows us to make disease models and so on. I think one area it's starting to develop with flies is to use them as mini humans. We call it humanizing. So for example, the genome of the fly is not very different to our genome. We have the same genes. So it's possible to replace a fly gene with a human gene. And we could replace that fly gene with a disease human gene, one with a mutation. And the fly can then become a model system for testing drugs. We call them avatars. So we can very quickly grow up huge numbers of flies. Each female lays about 200 eggs. So you start with one pair of flies, and after a month you've got thousands. They're all the same genetics. They may be carrying a human disease gene. We could screen 1,000 drugs against all of them very quickly. In the clinic, people are starting to think about using this approach to identify the right drug for the right person. So personalized medicine often means people respond to different drugs even though they may have the same mutations. And in some areas like cancer, where time is of the essence, there's scope now to make a humanized fly of an individual's cancer mutation and screen it for various chemotherapy agents to find out which one might work best. And that can be done relatively quickly and then move back to that patient in the clinic. So from a basic science, there's huge potential, but I think the applied science, how we can exploit the fly to help human treatments, is enormous.
Thank you, Richard. I'm going to start bringing the podcast to a close. So I just want to say thank you for coming on and sharing your insights. That's been incredibly interesting and exciting. And clearly, there's a lot still to be done with fruit flies.
Well, thanks, Dave. I've really enjoyed it. And I think I'll just leave by saying one thing to the listener, that if you take nothing else from this podcast, next time you see fruit flies buzzing around your fruit bowl during the summer, please resist the temptation to squash them. Just look at them as smaller relations. Thank you.
Thanks, Richard. This has been the Brain Fix podcast. Any feedback on this podcast or for future content or any sponsorship inquiries, please send to the DNS inbox. That is dns@manchester.ac.uk. I'll repeat that, dns@manchester.ac.uk. And please use the word podcast in the subject line. Tune in next time for more neuroscience.